Okay, thank you. Okay, so we'll move on to our second and final poster taster slot. So this is another warm welcome to Helena Almqvist from the Karolinska Institute, and Helena's going to give us an overview of CETSA technology for high throughput screening approaches.
So, yes, thank you for having the opportunity to talk about our work, because it's a collaborative work. My name is Helena Almqvist, as you said, and I'm from, I work at Karolinska Institute, CBCS, Chemical Biology Consortium, Sweden. And we are a national infrastructure that support academic research groups in Sweden with chemical biology research. And our expertise lies within screening, assay development and screening, medicinal chemistry, and computational chemistry. And the title of my poster is, as you said, Screening for Novel Inhibitors of Thymidylate Synthase using CETSA, a high-throughput target engagement assay in cells. And this is a collaborative project with the inventors of the CETSA method. It's a group at Karolinska headed by Professor Per Nordlund. And Daniel is here in the audience today, and also have another poster that will that presents CETSA work, but I will get back to that. So, okay, what I will do here now is that I will take a step back and just briefly go through what CETSA is, the basis of CETSA, and then I will continue with a work that we did to screen format CETSA, and then a third part is when we did then a full screening campaign of a new target, the thymidylate synthase. So, So the basis of CETSA is the biophysical principle that proteins get thermally stabilized upon ligand binding. So here, as you can see, a native folded protein, when exposed to increased temperature, they will denaturate, unfold, and eventually precipitate and aggregate. However, if they are, if a ligand binds or a drug binds to the protein, it will get thermally stabilized and melt and denaturate at a higher temperature. So you see a so-called thermal shift. Thermal shift assay is nothing new. It's been used in the industry and research for decades. But the thing is that it's only been used on recombinant proteins, on pure protein samples. So here is the new thing then, when Daniel Martinez Molina et al. Published last year a paper in Science describing a proof of principle study to study this, to study target engagement in a cellular context or even on tissues. So by being able to do this, it can give you a lot more information than only the target engagement study. It can actually provide information about drug transport, tissue distribution, metabolic activation, so forth, because it's the drug needs to get into the cells and perhaps get metabolically activated and so forth. So it provides more information than only the target engagement. So, okay, but how is this done then? This slide is a schematic figure of the original protocol, how it was set up from the inventors. So the cells are treated with the drug or the compound, and then they are aliquoted in PCR tubes, whereafter they are heated in a PCR machine so that denaturation and precipitation occurs of non-stabilized proteins. And this is important. And then you need to lyse the cells, and this is done in the original protocol by a freeze-thaw cycle in liquid nitrogen. And then the samples are centrifuged to remove the debris and protein precipitates. So the only thing that remains is the soluble fractions, and that is then analyzed on Western blot. And it can be quantified, and you can plot the melt curves, and they look like this. So what you detect is the soluble remaining protein that are stabilized. So here you can see with a drug, you get this thermal shift. The protein are stabilized. And this window is then used. This is what we've used in the screen then. So remember this picture, this window here. So we choose one temperature that we then use in the screen. So okay, this protocol is quite time-consuming. It requires a extensive sample workup, and many different applications of CETSA might require a higher throughput. So then this project was initiated with us at CBCS to adapt a high throughput screen protocol for CETSA. So our requirements that we saw, two main requirements, was to move over to a microtiter-based format to be able to introduce automated liquid handling steps, and also to move over to a homogeneous assay detection format. So we didn't need to separate the soluble from the precipitated proteins. We can also then go down in volume and reduce reagents and stuff. So okay, and based on our knowledge and our expertise, and we also have the available equipment, we choose to go for alpha screen. There are lots of other homogeneous detection assays out there, but we used alpha screen. And for this reformatting work, we used test cases, two test cases. It was P38 and ERK, because PerkinElmer provide surefire kits, on-the-shelf kits, already standardized for this. And this work have been recently published in Nature Protocols, and Daniel is actually having a poster on that work here. It's number 144, I think. So if you're interested in this validation work, then we compare the original protocol with data using this new screen format protocol. And the outline, how it looks now, the screen format is this busy figure. So I'll step you through it. So what we do now is that we have moved over to a 384 well format. We spot compounds using an echo non-acoustic dispenser directly in the PCR plate. Thereafter, we add cells using a compound handling device. And then we incubate the cells for a while, whereafter we heat them in a PCR machine, a light cycle or equivalent. Then instead of the freeze-thaw cycle in liquid nitrogen, we use just ordinary lysis buffer, automated additions. And then we need to transfer some of the lysate to a detection plate. We tried to use the same plate. We tried to read in the PCR plate, but that didn't work. Still working on it, to be able to skip that step, because you want to minimize the steps in a screen assay. But anyhow, after that, you dump in all the reagents, antibodies, donor acceptor beads, and then you read. And I didn't say that for those of you who don't know alpha screen. It's a proximity-based luminescent assay. So to detect the protein in your solution, you have two directed antibodies directed towards two different epitopes of the protein, and then you have the beads that are linked to these antibodies. And if they come in close proximity, you get a signal. So now we established a really nice high-throughput screen protocol, as I said, just recently published. But we also wanted to do was to do a full screening campaign. We wanted to push it even further. So that's when we did the work on thymidylate synthase. And that's an enzyme that the group have deep knowledge of. And it's a homodimeric enzyme involved in the early stages of DNA biosynthesis. It's well characterized. It's got at least two small molecular binding sites and many known inhibitors out there. And it's used in clinic today. It's a known anti-cancer target. And this is what my poster is about. So now I'm finally to where I'm presenting on my poster. But there are no kits ready for this enzyme. So we had to design a new assay from scratch. And I'm not going to present all the details how we did it. Please come and have a look at the poster and I'll show you. But we had to design a new alpha screen protocol. So the screening for finding a new antibody pair, I'm presenting on the poster. But we did that. We set up a really nice assay for that. And this is the final result of the screen. We screened 10,500 compounds in a day. It's equivalent to 32 384 well plates completed in a day. And this is a scatter plot with the results of the screen. And as you can see here, we have a nice separation between positive and negative control. And negative control is a DMS, though. It's unstabilized protein. And the positive control is Raltitrex. It's a well-known TIMS inhibitor. and all the blue dots in between are the hits from the library. And we were really glad to see that we picked up many of the known reference inhibitors, and there are, as I said, it's 2 different sites. It's folate analogs, and it's the prodrugs, or the fluorouracil, for example, 5-fluorouracil. And that's a prodrug that needs to be activated, needs to be metabolized. So these hits, and this is a cousin of it, as an analog, floxuridine. These would not have been able to be picked up in an ordinary thermal shift assay on pure recombinant protein. So we're really glad to see that we have, this is just an example of a few of the hits. We have done confirmational studies of all the hits in dose response, and so we just did this before the summer, so we're still ongoing. We're looking through the hits and see. But there are lots of, as I said, reference inhibitors and analogs thereof, of both substance classes. So yeah, to sum up, just to say that we have gone from the original protocol, and together in collaboration with this group, we have now done a screen format of the CETSA work. So thank you very much. Keep that figure.
Thanks, Helena. I mean, that's a really nice approach to potentially look at proteins in their native settings, yes, rather than recombinantly purified enzymes.
Mm-hmm.
Take any questions, please. Time for a couple of questions.
Mm-hmm.
So could you comment on how quantitative the TM measurement is? Are you concerned about false negatives from your screening campaign?
I mean, since you measure the protein that are still left in the solution after heating, and you know that if the protein is not stabilized somehow, you will not have it there. So it will be there because it's been stabilized. So I think that you can say that the false positive rate is quite low. It's really, really low.